All cells have the same DNA, but several different factors affect how that DNA is utilized in different cells. We're going to start gene expression with some classic genetics and then talk about some newer discoveries. The making of a protein requires transcribing the information from DNA onto RNA and then translating the RNA into protein. Both transcription and translation are based on a triplet code in DNA and mRNA. mRNA, shown here in red, is matched to the template strand of DNA via transcription. Let's look at this in some more detail. Here's the gene anatomy we saw in the previous lecture. Remember that the non-template strand is labeled 5' to 3' and the template strand is labeled from 3' to 5'. Sometimes the strand is called non-template and sometimes it's called coding, but that's the same thing. There's no real top strand or bottom strand in DNA since it's really a coiled mess of chromatin all twisted together around proteins. It doesn't appear as stacked bars inside the nucleus. That's why it's important to look at the strand ends to decide which end is going to be the template. If we look closely at a promoter, we can see that it begins with a series of nucleotides of A's and T's. Because the nucleotides on the non-template strand are called TA-TA, this is often called a Tata box. First, regulatory proteins called transcription factors bind to the promoter to turn on the gene. Then, the enzyme that builds the mRNA, called RNA polymerase, binds to the transcription factors and the DNA and can begin transcription. The correct transcription factors are required to turn on a gene. This happens in many different ways in different cells, but here's an example that we've already talked about in intracellular signaling. An external signal molecule can cross a plasma membrane and bind to an inactive transcription factor. This creates an active transcription factor, which can now enter the nucleus and bind to a specific promoter. Once transcription factors bind to the DNA, RNA polymerase can bind as well and begin making an mRNA transcript. There are three phases of transcription: initiation, elongation, and termination. Once we have the RNA polymerase bound to the DNA, initiation begins. The important steps of initiation are that DNA unwinds and that RNA synthesis begins on the template strand. Notice that the 3' to 5' strand is the template strand used to make the RNA transcript. During elongation, the polymerase moves downstream, unwinding DNA, and the RNA nucleotides are continuously added to the mRNA transcript at the 3' end of the mRNA. After the RNA is transcribed, the DNA reforms its double helix. Then, at termination, the RNA transcript is released and the polymerase detaches from the DNA. How does RNA polymerase make the RNA strand? Here's a close-up. Notice particularly the following. Added RNA nucleotides are the complement to the DNA template strand and are the same as the non-template or coding strand. Again, the DNA template strand is being copied 3' to 5', and the RNA strand is being made 5' to 3'. Lastly, RNA does not have nucleotides with the base thymine. Instead, it uses the similar base uracil. Once an mRNA transcript is made, it's modified before it leaves the nucleus. One step is the removal of segments of the RNA. Removed elements are called introns, while the remaining elements are called exons. Only exons remain in the mRNA after this processing. Next, the front or 5' end of the mRNA has a modified guanine added, called a cap, and a long series of adenines are added to the end of the mRNA at the 3' end, called a poly-A tail. These new ends seem to serve the following purposes. They facilitate the mRNA out of the nucleus, they prevent the RNA from being digested by enzymes in the cytoplasm, and they guide the binding of the mRNA to the ribosome. Once modification is complete, the transcript is considered to be mature mRNA. This completes our basics video for today. Thanks for watching.